Hello, I'm Mark Taz. I'm a Principal Scientist at Waters Corporation, and today I'm going to tell you about the work we've been doing towards cellular resolution DESI, so particularly low flow DESI high resolution. So at ASMS 2023, Dr. Emrys Jones first introduced the concept of low flow DESI for high resolution. This was a non-commercial setup where we were using a cadent-free IMI Norgine syringe pump and some modifications to the source. So one of the jobs I was tasked with this in last year was to look at whether or not we could perform the same low flow DESI high resolution imaging using a commercial setup that the majority of our customers would already have. So this is the commercial setup that I was using. In this case, we chose the Xevo G3 QTOF mass spectrometer, the Acuity M-Class Micro BSM for solvent delivery, and then obviously the DESI XS source. Now the critical thing to get the low flow high resolution DESI is a very stable flow rate at sub 500 nanoliter per minute rates. Now this is why we've chosen to use the micro BSM, because this gives us a range of diagnostic information which allows us to see if anything is going wrong with our solvent delivery in terms of back pressure, pressure spikes, the pumping, which you can't see, say, on a syringe pump. This allows you to determine if you're having issues with your imaging because of your solvent delivery or because of something within the setup of the DESI source. Let's say we chose the Xevo G3 QTOF to look at mainly because of its superior sensitivity. So the G3 has a very high sensitivity, and with that sensitivity you can do a number of things. In this instance, we were looking at increasing the speed of the acquisition and seeing what effect that increase in speed then had on the data quality that was produced. So this is a brain section that's been scanned at 5 scans per second, 10 scans per second, and 20 scans per second. And what we did is we ran this through a segmentation tool to define regions that should have similar spectra between the different acquisitions. We then drew regions of interest across those tissues and extracted those spectra and performed a correlation. And what we found was that the correlation was very good. Actually, it was within what we'd expect for the normal biological changes within the same area of tissue, within regions of interest that were taken from the same areas. We can also see on the PCA plot that the samples separated based on their regions of interest, and we didn't see a separation based on the speed that they were acquired at, which tells us that that speed change is not really having an effect on the quality of the data or the end result. So what is the low flow DESI really? So this is a view of the conventional DESI. So typically with conventional DESI, we'd be running around about 2 microliters per minute. And this, with the current sprayer technology, allows us to propagate a beam that's around about 20 micrometers in diameter. With the low flow DESI, what we're doing is reducing that solvent flow rate, and this allows us to produce a much narrower beam around about 10 micrometers across. What we have noticed, however, is the propagation of that beam is much shorter. So this means our working distance is much narrower, and this is one of the things that we'll come across later in, which is quite critical. So the first thing I did was to go about looking at some optimized settings, so to give us like a reference of where to begin. What I found was a sprayer angle around about 70 degrees worked quite nicely. Now, when we first started doing this, we thought the gas pressure was going to be critical to getting a stable beam onto the target. And so we thought we might have to do a lot of optimization with the gas flow. So we actually moved the gas regulator outside the source. However, after going through multiple optimization routines, I discovered that actually pretty much all the time I was coming back to the same gas pressure around about 1.35 bar. What this means is now I can put that gas regulator back inside the source and recommend that people just use that gas pressure as a starting point. In terms of solvent flow rates, we've looked between 500 and 250 nanoliters per minute, and the capillary voltages tend to be around where they would normally. Maybe they've got a slightly narrower profile in order to get good ionization. The main considerations for running these experiments, slide height. This is where that distance that the spray propagates becomes important. So we have to get very, very close to the surface. So you have to be careful in variations between 2 different samples because you can hit the surface, and I have in fact scraped entire tissues off of the surface at times. Another thing I found that was useful was to allow there to be a source equilibrium around about 15 to 30 minutes after you load a slide. This just allows all the temperatures to settle, all the airflow to settle. And then finally, sample dryness. The slide needs to be visibly dry. So you can do this by desiccating it, or what I do is I just place it in the source for those 15 to 30 minutes, and the natural ambience in the source will dry the tissue to a suitable level. The critical requirement that we found was the back pressure from the pump. If we do this as we would normally, if we just put some tubing straight up to the DESI source, we get a back pressure around about 20 psi, and the micro BSM doesn't like that. So what we've done is we've patched in an Acuity C18 column to the solvent line and then replaced some of the tubing, and this allows us to get our back pressure up. So we found that really we need a minimum back pressure around about 300 psi for a stable flow to propagate. So in terms of actually optimizing the sprayer, the main optimization parameters are the sprayer height, capillary height, sprayer to inlet distance, and then the capillary voltage. And the sort of routine that we'd go through when we're setting this up is to set the inlet temperature, then set the inlet height, adjust the voltage to achieve a stable ion beam so we can see that we're getting some ions through. Then move the sprayer as close as possible to the surface, then adjust the sprayer to distance capillary. So we'll get the sprayer as close as possible, then move the sprayer forwards and backwards until we get a suitable signal response. Once we've achieved that, we'll fine-tune the voltage to achieve the maximum signal possible, and we might iterate on that if necessary. The initial results we got, so this was with 500 nanolitres per minute flow rate, so this gave around 700 psi back pressure, were not exactly encouraging. What we can see here is that the 500 nanolitres per minute flow rate, we've got an increase in image fidelity going from 50 micrometers to 20, but beyond 20 down to 10 and 5, we're not picking up any particularly more detail within the image. So we're not getting more information by having more pixels. However, if we halve that flow rate again down to the sort of 250 nanolitres per minute flow rate, which gave us around about 300 psi, we can now see that as we progress down in pixel size from 50 to 20 to 10 to 5, we're actually getting more information. We're seeing a better clarity of image. We're not just seeing the same thing but a bit blurry. Now, as I mentioned, the G3 is a very sensitive instrument, so we can go quite fast. But even so, with the increasing resolution, as we increase the resolution, it's a square law rule, so the amount of time it's going to take us to acquire these images increases basically exponentially. So even going fast, it's not necessarily possible or viable to do an entire tissue to gain the information we want. So what we did is we developed what we call the HDI microscope workflow. So this is part of HDI 1.8. And we're utilising a characteristic of DESI that DESI is a very kind technique, therefore we can go over an area multiple times, particularly when using the low flow DESI. So what we're doing here is we'll do a first low resolution fast pass with the low flow rate, which causes minimal tissue damage. This allows us to get a basic image of our tissue. We can then look at that data that's been generated and draw regions of interest that we want to go back and reacquire at higher resolution. So you see the boxes here, these are the areas that have been targeted for reacquisition. We then load those back into the HDI method creator and then increase the resolution and then reacquire those specific regions. And what we can see is it doesn't really seem to make a difference if we've done the pre-scan. When we look at these two images, the slightly bluey image is a fresh sample area, so that's not one that's had a pre-scan on it. The second area that's yellow has had a pre-scan at 100 micrometers, and we can't see visually within the image any evidence of that 100 micrometer pre-scan having been occurred. And also when we look at the intensities of the spectra that are generated, they're actually pretty similar. So now for a slightly more interesting sample. So that previous one was just some porcine liver. So here we've applied the same technique to look at a rat brain section. So we performed the whole sagittal rat brain at 100 micron and then targeted key areas of interest that we wanted to go back and look at. And here we've got a 5 micron view of the cerebellum. We can also see here that we've got very good alignment between the dataset coordinates and the dataset base coordinates for the two images. So one of the other things that the microscope mode is quite useful for is for trickier samples. So this is some previous data that we acquired in 2022. This is a mouse adrenal gland, and we were actually quite very happy with this image at the time. What you can see here is we've got an outer layer, so the capsid, then we've got a cortex and medulla within this tissue section. So you've got sort of 3 regions. Now what we'd expect is to see multiple regions coming in within this tissue. Now these samples are actually very challenging because they're very small. They're also very difficult to section. These, in fact, were the best sections that we got from the previous study. So we thought, well, we'd have a go at doing this again, but now at the higher resolution of 5 micrometers. Unfortunately, the section didn't go as well this time, so we haven't been able to get a full cross-section of an adrenal gland. Also, because these need to be embedded and they're very small, these were within OCT, which makes it very difficult visually to identify exactly where the tissue was within the sort of embedding media. So we used the precursor scan just to identify where we were seeing lipids, and then we went back and re-imaged this at 5 micrometers to create what I call the pizza slice. Unfortunately, we don't appear to have all of the tissue here, so I think what we're looking at is sort of the outer layer and then the secondary layer. But this proved as a proof of principle, and so we're working with Sheba Jarvis at this point, and we decided to go ahead and have a look at how robust this technique would be in terms of sort of more large cohort analysis. So here what we've got is a series of human adrenal tissue. So this is a mixture of different cancers present. Some are secreting hormones, some are not, some are benign, and some of this is actually normal tissue. So we did an initial sweep of all of these tissues at 50 micrometers just to show that we could get valid data using the low flow mode, but at sort of not high resolution imaging. And so we took all of these, drew regions of interest all across them, and performed unsupervised PCA. This allowed us to pull out a couple of ions that would show different distributions within the tissues, which gave us this lovely view. So then what we did was to target regions of interest across a few of these different tissues. So I'm just going to show examples from these 3 tissues here, where we could go back in and look at areas of interest where we could see differences in these 3 molecules. So we selected samples 3 to 5, and we drew a series of regions across these. So this is sample 3. And these are the results of the 5 micron rescans of those selected areas. And then this is sample 5. And then the one I thought was prettiest was actually sample 4. So what we've now done taking this data is we wanted to have a look and see what was going on at this sort of higher resolution. So we performed segmentation mapping using the UMAP segmentation. This kind of gives us an idea of the different regions that are present within the tissue. We then can overlay that view with the original 5 micrometer tissue to try and work out what regions are what. So this is work that's still ongoing. We're awaiting the results of Ki-67 immunofluorescent staining. This should show us areas that are proliferating. We want to see if any of those areas that are proliferating match up with areas that looked interesting that we reacquired at 5 micron. We then should be able to pull out spectral information about those regions and why those regions are different. So I mentioned that you probably wouldn't want to do an entire tissue, but I did have a weekend where the instrument was not being used. So I performed a 10 micrometer pixel size image. And this is kind of the numbers here that kind of give you an idea as to why you might not want to do this. So it's 10 micron pixel size. Originally I was going to do a 5 micron image, but then I did some calculations and I worked out that it would produce around about 1.5 terabytes of data. When I looked at the hard drive on the computer, I only had 750 gigabytes left. So I decided to drop the resolution a bit. So what we actually ended up here with is a data set that's 364 gigabytes in size. This is my record for the largest image I've ever produced. This took 35 hours to complete, even though it was done at 20 scans per second, and resulted in a 14 gigabyte processed file, which is also the largest file we've ever tried to put into HDI, and I can say it's successfully loaded. So we've got over 2.3 million pixels that displayed correctly. I also performed continuous log mass correction on this data, and we found that the mass accuracy error was better than 2 ppm. across the entire tissue. And this just shows the results for the 798 peak with a 10-minute sampling frequency and a 2-minute sampling period. What we can see is we're nicely distributed around about the 0 ppm error, and there's no significant trends or shifts in this data. And it's not just one or two images that we can get out of this data. So here we've got the visualization of the 20 most abundant monoisotopic tissue analytes. So we decided to investigate this data a little bit in more detail. So in this case, I've just taken a region of the data to make it a little bit easier to handle. So what we're seeing here is the 100 most intense ions displayed to sort of create a pseudo TIC image. We took this image into the segmentation mapping tool and had a look at what we could see. And what we saw was something quite interesting, was this little purple speckles here, which seemed to localize down to only a few pixels across each. So as we look at this more detail, we look at microscopy image in more detail, we can see that these little clusters within the segmentation map seem to indicate these small cells that we can see within our brain matter. And having a look through the different ions, I managed to find a 760 peak that matched the distribution of these. So at the 10 micron imaging, we can see that in the sort of lower region, we're not differentiating the cells. So we can see the cells are differentiated, but in the lower region, they're not differentiated in the segmentation mapping. And then within the 760 image, they're kind of starting to merge together. So what we did was to have a look and see what happened if we then stepped down the pixel size to 5 micrometers. And at the 5 micrometer, we saw an increase in fidelity. So now suddenly we could separate these individual cells. And from these cells, we can start to do some estimation of the actual resolution that we're achieving. So DESI is a convoluted technique. So we're acquiring ions from the complete area of the solvent beam. So if we were to do a pixel at 5 micrometers with a 5 micron beam, the best resolution we would expect to get would be around about 10 micrometers. And actually what we're seeing here is we are achieving that. We're starting to see a resolution of around about that 10 micrometer level, because from the microscopy image, I've measured the cells to see that they're around about 10 to 15 micrometers across, and we're getting 2 to 3 pixels across the tissue. So actually, chances are the desorption area is less than that 5 micrometers. So I mentioned that the back pressure is really important. So at the minute, we are using a column to try and generate that back pressure. So now what we're doing to go forward is to look at alternative ways of generating that back pressure. So one of the ways to do this is to use a back pressure regulator. So here we've got a chart where it shows the different regions. So we've got the sort of low flow region, which we consider sort of sub 500 nanoliters, and the normal flow region, which is above the 500 nanoliters. On this chart, we can see at the bottom, this is if we use the tubing only, and we're getting pretty much no pressure at all. With the column, we're getting the sort of 250 to 300 psi at the 250 nanoliter flow rate. However, if we use a back pressure regulator, we can push this well up to above 1000 psi. It also gives us a relatively flat profile of pressure across the entire range. So it might actually be beneficial to the normal DESI routine as well. And this is just some of the very, very initial results we've got. We haven't got much data on this yet, but this is performing the same sort of imaging on liver using the back pressure regulator. And we can see that, again, we're getting this transition in terms of the resolution fidelity as we go down in size of pixel from 20 to 10 to 5. And finally, I mentioned at the beginning of the talk that we were looking towards cellular resolution imaging. And I think I've shown a little bit of that with the cells that we could see within the brain tissue. However, this is some late-breaking work that's been done recently. So here we're looking at cells grown on glass slides. So these are PC3 and differentiated THP1 macrophages. So each one of these blobs is actually an individual cell. And these are around about 10 to 15 micrometers across. And we've imaged these ones at 5 micrometer pixel size. And we're seeing pretty much that sort of resolution that we'd expect. And when we take spectra from each of these cells, we can put these into statistical packages, and we can see differentiation in the lipid profiles. So in conclusion, the main question was, can we apply the low flow, high resolution DESI method to a commercial setup? And the answer was yes, we could. Was it difficult? Surprisingly, no, it wasn't. The key requirement to getting this to work with the commercial setup is to develop good back pressure for the solvent delivery, and we can achieve a resolution estimated at less than 10 micrometers. And we're currently looking at alternative solutions to generate back pressure, aiming to achieve that better than 1000 psi at 150 nanoliter flow rate. Thank you.